Gedalanga, who is an assistant professor in the Department of Health Science here at Cal State Fullerton. He earned his PhD in Environmental Health Science and Policy from the University of California, Irvine, and was a postdoctoral scholar at the University of California, Los Angeles. His graduate and postdoctoral training established predictive links between microbial populations and water treatment outcomes. Currently, Dr. Gedalanga is investigating the response of microbial communities to emerging water contaminants and other pollutants to determine the greater impacts to environmental health. His teaching and research interests primarily focus on the interactions within ecosystems that balance health risks, ecological function, and environmental policy. So let's give a round of applause.
So thank you for that wonderful introduction. That was very comprehensive. It really defines who I am and where I've come from very accurately. I've always had this component of water in my studies and in my research interests. So it's something that I've connected with very strongly, and you can see it throughout my education. Now, I wanted to talk to you all about environmental health, but before we go into that, I think maybe I'll just talk a little bit about my experience, since this is kind of like that taste of graduate school, and maybe this is something that you've heard from other faculty members as well. But one thing that I think is wonderful is that you're all already thinking about graduate school. This is something that you guys are actively looking to be involved in. Now, how many of you guys are juniors or seniors? Are any of you guys graduating? How many of you thought of graduate school maybe when you were a freshman or a sophomore? Yeah, see, myself, I did not think about graduate school until the last quarter of my senior year. Now, that's a very late thought of getting into graduate school. In fact, I actually took a year off. I did research in a lab before really committing to graduate school, but I didn't even think I could do it until I started that last semester. Not even if it was a quarter. We were on the quarter system, we're on the 10-week system. So I didn't even think about it until then. And it's, thinking about it at that stage of your higher education is almost too late, but it's never too late to start something. So the fact that you guys are all here and thinking about it and finding ways and getting that path towards graduate school, it is a wonderful thing, and it's going to only make you better prepared as you make that choice and start applying. But I went to graduate school. I did all my education here in Orange County. I was undergrad at Irvine, at UCI. I also did my PhD work there, and my research was always with water. For my graduate degree, I studied both microbial source tracking, which is this article that we're going to talk about today and discuss today. Which, be honest, how many of you guys read this article or got through a little bit of it? A couple of you, maybe. Okay, and that's totally fine. I didn't expect everyone to really read the article, so we're going to talk about the article in the lecture, just kind of like walk you through what was being done in this article. But I did microbial source tracking in my graduate degree. What that really is just trying to identify sources of pollution from in an impacted water body. So say we go to Huntington Beach and we want to, we're thinking about, do we enjoy the water? Of course we're going to want to get in the water, but are we going to be negatively impacted by whatever microorganisms are in that water? And if we are, what is the source of the bad, microbial organisms that are in that water? So we were trying to figure out a way to tell you whether or not it was safe to get in the water, and also what were the things that were contaminating that water. So that's what I did in graduate school. When I finished graduate school, I was not quite convinced where I was going to go. I wasn't sure if I wanted to stay in higher education as a faculty member, or if I wanted to pursue more of a private industry type of position as a consultant or as some other type of career. So I ended up doing what a lot of people do in that position, which is do a postdoc. And actually, a postdoc gives you an opportunity to do two things. You can either branch out into other areas that are new and exciting in terms of like your research interests, or you can buy more time in a sense in just studying like some of the same things that you've been doing. Now, with research, there's never a final endpoint. You know, there's always a question that you end up, you know, inevitably coming to as you continue your studies. So what I chose to do was actually move away from microbial source tracking and move into bioremediation. So how many of you know what bioremediation is? A little bit of an idea. So bioremediation basically is using bacteria to clean up a contaminated source of or its contaminated environment, whether that environment is soil or water or air or whatever. So I would look at how do I clean up this water contaminant, this groundwater contaminant, with the bacteria that were already there. So we had a study of, you know, a lot of different things. We were interdisciplinary. I worked with environmental engineers. I worked with environmental microbiologists. And it was a great experience because I was able to interact with this diverse group of people and still, you know, continue to research some of the things that I really enjoyed in that particular area was how do I use microorganisms to our advantage? And you can see there's many ways you can clean up a contaminated water source, right? It's not only just using bacteria or fungi or any other type of biological organism. You can do active approaches, things like add chemicals or things like filter things out or absorb them onto, say, a piece of carbon or something like that. And what the advantage of me studying the bacteria is it gave me more of a sustainable option, right? So you think about bioremediation, using bacteria that are already there or bacteria that you add to an environment and really come up with a sustainable solution because you're no longer having to spend money in buying chemicals to drop into the environment. And you save the environment from those same chemicals that you're trying to use to clean up other chemicals. Sometimes when you add chemicals to the water to clean up one contaminant, you actually create another contaminant. And that's something that, you know, we just want to avoid. So I did that for my postdoc experience, and that was an extended experience, but that inevitably led me to Cal State Fullerton, where I get the opportunity to talk to you about environmental health because this is something that I really connect with. How many of you know what environmental health is? Any environmental health science track people here? No? That's great. That's a good thing, right? Because now we all get to learn something new, but we get to understand where environmental health really lies, maybe in your potential career paths. Now, I know you guys are not going to be an environmental health scientist or anything like that, at least not at this moment. But let me just hear some of your backgrounds. Any of you in nursing? No nursing? Health science. I heard health science earlier. Most health science? What focus area in health science?
For me, it's global health.
Global health?
Mm-hmm.
Community health.
Community health?
Global.
Global health. And so you can see that both of those, global health, community health, all have an environmental component to it, right? So if I were to ask you, was there an environmental component in something that you really enjoy about global health or community health? Can you identify that? Yeah?
Yeah, I think most recently when I went to Costa Rica, that country is really big on eco-friendliness and conservation. So just seeing how everyone's mindset is like, oh, we need to make sure that we recycle and that we're using like only 10-minute showers or something like that.
Right. Really, this whole emphasis on preserving your natural resources, right? Because if we start to let those resources, decline, then we will eventually come up to a situation where we're now combating for natural resources. We might actually contaminate these resources, and it can impact our own health. Now, outside of health, are there any other majors out here outside of health science?
Human services.
Human services. Okay.
Kinesiology.
Psychology. Now, I mean, every one of these has like an environmental component. Now, it's outside of my specialty area. I'm more of a, how does water impact our health? But, in other things, there are ways where we can look at how the environment actually impacts our health directly, not just the water, but maybe the climate, maybe it's like the geography as well. But I'm going to talk to you about this definition by the American Public Health Association. They say that the branch of public health that focuses on the relationships between people and their environment. And that's, you know, really at the core of what environmental health is, and really at the core of what I really enjoy studying. I want to put that in a different kind of framework here. We like to kind of describe this as upstream environments with downstream health issues. This chemical compound may be, you know, completely foreign to everyone in this room. This is what we call perfluorooctanoic acid. Now, it's a crazy looking compound, but there are some things that you can see here. And I'm going to use this compound as an example to explain what is upstream environments, downstream health. So this compound, PFOA, P-F-O-A for short, so you don't have to write down this whole name here, is made up of different carbon-fluorine bonds. You can see C and F up in this thing. And then it has a carboxyl group at the end here. The big thing here is this carbon-fluorine bond here. Now, carbon-fluorine bond is a very, very strong bond, so it's really hard to break up. So when you have this particular contaminant in the environment, it's very, very difficult to break down. What if I told you that each one of you were probably exposed to PFOA? Would you believe me? Maybe. You don't know where it's used, but you believe me because I'm very trustworthy. Now, it's actually used in many different products in all of your households that can be found. PFOA has also been used as a non-stickware on your pans. So if you use non-stickware Teflon-coated pans, you might have an element of PFOA in it. Now, just because it's mentioned and used in a lot of different things doesn't mean it's extremely toxic. Now, the toxicity on it is still being studied. It is classified as a possible human carcinogen. So we want to avoid contact with it using the precautionary principle. Like if we kind of know that it has a negative component to it, let's try to avoid it at all costs. The United States has not quite made that distinction yet, but we start with this one chemical. We know it's distributed in our home environments, and we can see that each of these different products might have it. You notice the fire extinguisher, PFOA and perfluoro compounds are present in fire extinguishers because they're very good at putting out fires. We might see it in plastic bottles, paints, lacquers, and in all of these different household products, especially on your pans. And that's why I say every one of you has been exposed to this particular compound. Now let's think beyond that, right? So this is our homes. This is just like one little area. How does it really become more of an issue? Why don't we just get rid of this in our homes, right? Well, you can see that it's more than just our homes. Once it gets out into the environment, because it's such a strong bond, it can actually persist for very, very long periods of time. Now, if we just did a blanket study of where we find PFOA or PFOA, we see that it's pretty much ubiquitous in the environment. So what does that mean? It means we find it wherever we really look for it. Now, we can see that it happens to occur in wastewater treatment plants. So maybe the waters that come from our home are bringing PFOA into the wastewater treatment plant. But it doesn't just stop there, right? Because once it gets to the environment in the wastewater treatment plant, that water gets distributed in other environments. So it can be something as complex as the water cycle, where you have all these different paths and routes of transmission in different water environments. So here's the wastewater treatment plant. We find it in foods, we find it in soils, we find it in metropolitan areas. Can you believe we even find it in polar bears, right? So what is the significance of that? Let me just pose that question to the group here. What's the significance of finding it in polar bears? Like, what is a question that you might have to follow up knowing that you can find this thing in polar bears?
Are we talking about on polar bears or
in their bodies? In their bodies.
That means they've been drinking a lot of water. I mean, they are around like ice and whatnot. So, especially since they're exposed to it all the time, and then they play in it, they lie around with it, they sleep in it.
Right. So what you're thinking is, well, I wonder how it got in there, right? Mm-hmm. You're wondering, and you're justifying it by saying, oh, well, maybe they're drinking a lot of this potentially contaminated water, right? And one of the roles of an environmental health scientist is to really understand how things are distributed in the environment and then how that might eventually come into contact with a human population. So if we look at the polar bear, we know that this particular compound, PFOA, is man-made. It's not found in the environment naturally. So it's not part of the Arctic environment where this polar bear calls home, right? So the bigger question then becomes, well, how did it get there? And that's something that we like to study, something that we like to identify and find out. We know that this compound just travels throughout the environment because it can persist so long. It can get into the atmosphere, it can travel through the air. And so that's how it's getting into all these wide-reaching places. Now, we don't like to just stick to the contaminant alone. And in my area, I look at waterborne diseases and waterborne contaminants and those types of things. And so we're going to switch now to the actual journal article itself. You may have a copy of the journal in front of you. And I know not everyone had a chance to read it or look over it, but this article talks about water pollution and how water pollution can be a public health concern. This is like the real, like the bigger picture of this particular article. The title alone, Mobilization of Microbial Indicators, Microbial Source Tracking Markers, and Pathogens After Rainfall Events. That kind of speaks to a number of different issues in the emerging water pollution issues. And so that's kind of why I chose this article for you guys to be exposed to. Whether or not you get an opportunity to read it, I hope that maybe you think that it's interesting enough to go into the details and read this article. So one of the things that I hope to incorporate into this particular short discussion that we're going to have here is enough information to really understand what the article found or what the article was doing in terms of the research that was conducted. So we have to start this off with a discussion about microbial indicators. Now, microbial indicators are used in order to identify whether or not that we have a health risk from being exposed to this particular water. We can't just measure all the different pathogens or other different types of harmful elements that might be in a water source. It's just impossible, right? We can think of thousands and thousands of different types of pathogenic strains of organisms, and it's just not feasible for us to monitor all of those things. So one of the things that we like to do is we like to try to identify an indicator. This is one of the ways that this is done. These microbial indicators are actually present usually in any type of water environment, but their increased abundance really suggests that there's a potential hazard to human health in that particular environment. So any of you guys take biology or microbiology and do some Petri dish plating? But this also, for those of you who haven't, you probably have seen something like this before, right? Like it doesn't look totally foreign or unfamiliar. This is a Petri dish that a bunch of different bacteria have colonized onto. We've taken a water sample, we filtered it, and then we try and grow whatever organisms can show up. Now, the EPA has mandated that if we have more than 126 of these colony-forming units, is what we call them, on a plate, and those are all E. coli, then that's actually in violation of our water regulations. So we would say, don't get in that beach, don't get in that river, stream, whatever it is, don't get in that lake, because it has an elevated microbial count, and that means there's probably pollution associated with it. We have different types of indicators, and we'll talk about that throughout this lecture. Now, even though we monitor, and even though we have very good water standards, we still have outbreaks here. How many of you have heard of recent beach closings and other types of things like that, or other types of water outbreaks in the environment? I mean, this stuff happens all the time. Well, not all the time, but frequently enough that it has a very important public health angle, and we want to try to address some of these things. You guys remember that hepatitis A outbreak down in San Diego, or heard about that? These are some of the water issues that we're very concerned about. They do happen, and so we want to try to find ways to avoid them. The other thing that this article speaks to is this issue of pollution, and we break down pollution by two types. We call them point source pollution or non-point source pollution. Now, we want to be able to differentiate between these two. A point source pollution is basically anything that's coming out of a pipe. So if we have a wastewater treatment plant, they have a pipe that discharges into the ocean. If you're familiar with how it works here in Orange County, some of the wastewater treatment plants discharge about 5 miles out from the ocean, and that goes into the ocean. The thing about point source pollution is we can monitor it. We can identify whether or not there's a lot of bacteria or if there's a lot of chemicals, and if there is a problem, we could stop it and we can, make a change in treatment and address that. Non-point source pollutions are a little bit trickier. And these are things that you can find anywhere in the environment. But the reason why they call them non-point source pollution is because we can't exactly identify the particular source of pollution. Usually this is associated with runoff. Okay, so like after a storm, we know we don't want to go surfing in the ocean. We don't want to, go boogie boarding. We don't want to really play in that water or recreate in that water, because after a storm, all the water in the urban environment and in the mountains and all gets collected and the runoff goes into the ocean, right? So that's one of the reasons why we don't want to recreate after a storm. And that's because of the number of different non-point sources of pollution that can be carried to the ocean. All right. So again, we don't want to be swimming whenever we get a storm event. And one of the reasons why this study took place at this particular area is because it is in an urban area. If you did look at the article, you'll know that this area is in Spain. So this is not in the United States. This is in Spain, near the city of Barcelona. And it's actually looking at the Llobregat River, which I'm probably not saying correctly, and I apologize for that. But the Llobregat River is this river that runs through here. And one of the reasons why I have this map is because I wanted to show you that it's a river that's close to a heavily populated area. So Barcelona is a city of about a little over 2 million people. And when you have a very heavily populated area, you have to be able to treat the water, the wastewater that comes from all those people. And so that wastewater eventually gets dumped into the river and discharged out into the ocean. It doesn't get just dumped in. They treat it to a high quality standard. But you can see that if you have a very pressing demand, increased population, more waste, then there is a potential for some contamination to occur. The other thing is you can see that there's these mountainous regions, right? So anytime it rains, there's going to be all this water that's collected. It's going to filter down to this drainage basin. Now, it's about 110 miles, this length of the river, so it covers a large area. It's about the distance from here to the US-Mexico border. It takes about, close to 2 hours to drive down there, so this is a very large river. Nearest population is Barcelona, and it is heavily populated. So what did these guys do? These scientists, this research facility looked at different microbial indicators. They looked at E. coli, somatic colophages, sulfite-reducing Clostridia. It's a lot to handle there. But the biggest thing you really want to know or take from this particular information is that it looked at bacteria, which is E. coli. It looked at viruses, which are the colophages. And it looked at this other set of bacteria, Clostridia, which is kind of an anaerobic. So now we're looking at different types of physiological characteristics for the bacteria itself. E. coli is a facultative. It's an organism that can exist in oxygen and in oxygen-deprived environments. And it looked at human markers here. Now, these human markers, what are they? They are designed to identify whether or not the source of pollution is from human origin. Okay, so like we can look at a water sample from Huntington Beach, and we can say, well, if these human markers are there, then that means there's a human fecal contamination that's associated with that marker. Usually that's from a wastewater treatment plant, right? So because all of our wastewaters go to the plant that gets treated and then gets discharged. So we can say, well, it's because the treatment plant was in violation of something. Or it could be that there might be just other type of fecal contamination and we shouldn't be recreating in those waters. Then it looked at pathogens, known pathogens as well. So these are the indicators here. These are the actual pathogens themselves. So it looked at salmonella and also enterovirus, and those can cause human disease. So those wanted to look at some relationships between the indicator itself and the pathogen itself. So you'll also notice in the title, it looks at after rainfall events. And so that is a key component and a main study goal of this particular article, because that's going to look at the influence of climate change. And that's one of the identified goals. They also wanted to look at the efficacy of indicator organisms. That's just kind of a fancy way to say, how good does this indicator organism really work? It wants to look at environmental distribution. How do the indicators, how are they spatially distributed, or how are they more associated with one environment versus another? And also the persistence of those organisms. Do they stay around for a very long time, or do they kind of die off and then go away? We need to kind of know this information in order to be better informed and protect public health. So what are the things that they did? Well, they compared wet and dry periods. That's, to study the influence of climate change, they looked at what happens when we look at, before and after rain events. Because with climate change, we expect to see more frequent, or, depending on where we are, we expect to see different periods of rain, and we want to account for that. It looked at co-occurrence of indicators and pathogens, so that's how they determined whether or not the indicators were efficient and good at what they do. So will the indicator show up when the pathogen shows up? That's kind of the idea there. Determine the differences between sediment and water column samples. So how do the organisms associate with either the soil, or are they in the water? And one of the ideas here is that if it's in the sediment, it might serve as a source for future problems in terms of contamination. So if we have pathogens that kind of associate with the sediment, that means they're going to eventually settle down in the water column and they'll be in the soil part. But if there was enough flow, they might get resurfaced and redistributed into the water and become a problem for human health. And then they identified relationships dependent on the distance to the river. So they looked at areas in the water all the time versus areas that were just partially covered in the water. And that dealt with differences in flow, right? Because like the river, the level of water in the river is going to change over time. And they wanted to see if that had an impact on our microbial indicators. So they did a number of different things to actually count the indicators. They use two main types of methods. One is culture dependent and one is culture independent. Culture dependent method requires you to actually be able to grow the organism. Now there are drawbacks to that because we know we can't grow all organisms. We know that we have like a finite knowledge of how much of what these organisms can use for growth, carbon, nitrogen, phosphorus, and the media can be very complicated. So we can't really grow all the organisms. But if we use a culture-independent approach, then we're able to quantify all the different types of organisms in a water sample. Whereas a culture-dependent growth is only going to quantify the ones that can actually grow. And what happens if that particular bacteria is injured or not as happy as some of the other bacteria in that sample? Will it still grow? And that's a question that really hasn't been addressed. So these were the ones that could grow that they were able to grow, CEC, SRC. I'll briefly talk about them real quick. This is E. coli. This is the Clostridia. This is Bifidobacterium, total and sorbitol fermenting. So this one here is actually associated with humans or could be associated with humans. This one here is the coliphage, the virus, and this is Salmonella. Now we have culture-independent methods for total E. coli. This here is a human marker, Bacteroides, and this is enterovirus. So with the growth methods, you probably have seen something like this before, the Petri dish. This here is actually plaque-forming units, that's colony-forming units. This one is going to enumerate viruses, because the virus, this coliphage, actually feeds on bacteria, so that's why you get this clearing over here. This is a lawn of bacteria, and whenever there's a clearing, that's where the virus is supposed to have infected those bacteria. Then we count these plaque-forming units, we count those colony-forming units. The Salmonella was enumerated using the MPN method, and I want you to look at this particular method here because it's actually based on dilution. So we dilute all these different samples, and then we incubate it and see how much grows. And eventually, we might dilute out the organism of interest here. So we have like only 2 positive tubes here, where before at a higher concentration, we had all of them positive. And then eventually, there's no positive. We plug those numbers into a calculator, and then it tells us approximately how many organisms were in that water sample. MPN stands for most probable number. So you can see that that's not the most accurate way to count how many organisms are there. For culture-independent methods, anyone here heard of quantitative polymerase chain reaction, qPCR or PCR. If you've seen some of those CSI shows, you'll notice, hey, you know, let's do this forensic evaluation and see whether or not they were present. We'll do the PCR on the DNA, and then that'll tell us whether they're there. That's exactly what this is. So we know that there are specific DNA sequences that can be attributed to one or more types of organisms, and so we use that here for qPCR evaluation. Now, what were some of the results? These are the figures straight from the article itself. Figure 1A really looked at the water column and how wet and dry periods differed. So this here is the dry period in the open circle, wet is in the gray. And so we would look at this, you know, if this were you know, graduate level class, you would look at these results and you would really look and see whether or not you were convinced by the quality of the results, or whether or not that there might be misinterpretations of these results. The authors suggest that there's no difference between wet and dry sampling. So we look at just the water column, and you can see, you know, there's a lot of overlaps in some of these error bars, right? And so it looks like, I mean, just looking at it, I wouldn't be able to say with any confidence that there's any difference here. But they looked at it differently. They looked at what if we incorporate the flow rate as well. And so they incorporate the flow rate, the river discharge, and they said, well, when we do this, we can see significant differences with each of these different types of indicators that we looked at, except for the total E. coli indicator here. And you can see that, yeah, they definitely overlap. They don't look different at all, right? But other things here, this they called a significant difference. This they called a significant difference. They called everything else here a significant difference. Then they looked at the different sediments. Now, to give you some more background information, sediment A was the one that was in the water all the time. Sediment B was the one that was only partially covered in water after a rain event. Okay, so whenever they had a rain event, sediment B was covered in water, but under normal circumstances, it was dry. And they show here that there is actually differences in the way that they quantify the organisms based on the distance to the water. So you can compare the difference here between dry and wet period. Here you have about, we call this an order of magnitude. Here you have about a 1 order of magnitude, but in sediment B, we're looking at the same indicator, and we see about 2 orders of magnitude difference there, right? So they said that there is definitely a sensitivity issue there, except for these examples here with total E. coli. Total E. coli did not really have that same relationship, and then the bifidobacterium as well. Now, this is a busy slide here, but there's one key message that I want to kind of communicate to you all, and that is that microbial indicators were always larger than the pathogens themselves. So they were kind of doing their job, right? They're supposed to be in more abundance. They're supposed to be non-pathogenic so that if you find them there, you don't have to worry about being exposed to something like this. But they're also supposed to show up whenever there is the pathogen itself. And here you can see that the shaded area is really the area where the indicator was at, and then down below it was where the pathogens are at. So these are where they were enumerating pathogens. Whenever you see this dotted circle, that just means it wasn't present. So it was not detected. It was detected at, or it was below their detection limit. So they said that these indicators actually were very good at being present when there was a pathogen present. So the conclusions that they came up with were pathogens in the sediment are a public health risk. We were seeing these differences between the water column indicators and pathogens and the sediment indicators and pathogens. And a larger number were definitely associated with the sediment. So they said that pathogens in sediment should be recognized as a public health risk. The coliphage is a good indicator of enterovirus, and we should continue to use this for our regulatory framework. Total E. coli needs to be used with caution because you saw that there were no differences between the cultivable E. coli, and they did not vary. They did not co-vary. So when one went up, the other one did not always go up. Sometimes it went down, and that was a problem. And then they provide this ratio of coliphage to Bacteroides or human marker. So coliphage to human marker ratio of 2 is a good indicator. And I'm not sure why that doesn't show up, but it's basically a good indicator of human fecal contamination. So those were really the conclusions of that particular article. We talked about the results as we walked all the way through those results. These were the conclusions presented by the authors. And so I wanted to see if you wanted to discuss this particular article. How does this proceed after the article? Are we here to discuss the article itself? Yeah, so one of the things I wanted to find out is, what did you guys think of the article? What did you guys think of the data? If you kind of internalize some of the results, would you be able to kind of process it a little bit and provide kind of your opinion on it? Was this a study that you kind of think is important? Is there a relationship here between the research itself and the ultimate goals of protecting public health? Anybody? Anybody shocked at some of the results?
I'm not shocked because I actually took I have microbiome right now.
Yeah.
I just did water analysis this week. So a lot of the things you talked about really did hit strong. Yeah. Because it just reinforced what I learned earlier in the week. I especially got excited when you talked about most probable number because we specifically did that. In my microbiome class, we talked about, because I'm taking it at the JC. Yeah. So I'm taking it at Golden West. So we're near Huntington Beach right now. Yeah. So we're talking about how many microbes you need. So you need about 1,000 to 1,100 coliform forming units in order to close down the beach. And I was just super amazed earlier in the week, so it just reinforced it. Nerd. Yeah, so I thought it was interesting because talking about fecal matter, in my class we talked about how we can't really detect what fecal matter you can find because of E. coli, because there are other bugs, other animals dumping their
depositing their fecal material.
Yes. So I'm just interested, how can you detect whether the fecal matter is from a human being or from an animal or some other source?
Yeah, no, that's a great question, and that's something that those of us in the microbial source tracking world really focus on. We're there to, and it's kind of funny because it is, not dinner material. You don't talk about this over meals, but one of the things that we're really focusing on is how do we differentiate, between all the different animals that are out there and other sources. So the way that we do that, and the way that these researchers kind of use to differentiate is based on DNA. So we can look, now with MPN method, you're looking at E. coli or you're looking at some type of bacteria, right? With MPN method, there's no specificity at all. It's basically any warm-blooded animal that has fecal material that may have, contaminated that water is what's the source of those coliforms. You're talking about coliforms, which are found in all different animals, all different warm-blooded animals. But with the DNA, we can look at very specific organisms. Bacteroides, which is one of the organisms targets, that's that GA17 Bacteroides. From the studies we've seen so far, are very specific to human origin. So we can use that. We can look at the DNA, the 16S DNA sequence for that particular microbe, and then we can quantify that. And if it's present, it should associate itself with humans. Other people have looked at other things too. They've looked at different types of functional genes. So genes related to whether or not there's a virulence toxin associated with it. So one thing we looked at before was cow disease, right? So cows can be infected by E. coli that carry a particular virulence factor. And that virulence factor is only associated with those cows. So if you can find that virulence factor, the sequence for that virulence factor in your water sample, then you probably have a bovine or a cow source of fecal pollution. So you can look at very specific things with these molecular methods like DNA, and that really helps narrow down what is contaminating your water. So that's why they looked at so many different types of fecal indicators or indicator organisms, because they're really trying to figure out what is the source of their problem here. Yeah.
Another question.
Sure.
Is there a way of, I know there's probably a way to remove the bacteria from the water, but is there a way to remove like the enterovirus or any other virus from the water?
Yeah, so viruses are an interesting an interesting target here because we know there are differences between the way a virus can exist or persist in the environment versus bacteria. And oftentimes we see that viruses are very hardy and can actually persist for a little bit longer. So we're really looking at targeting these viruses. They can be filtered out from water. You can separate them physically just by physical separation. But of course, you think about how to do that with very large volumes of water, that becomes incredibly challenging, especially when you think of the pore size has to be very, very small. We're talking about 0.22 microns here because, viruses are extremely small, whereas bacteria are much, a little bit bigger than that. We can get away with a 0.45 micron filter. And so the smaller the filter size, then you're more likely to be clogged, and it makes it very difficult. The easiest way, though, is just to disinfect, right? So a lot of the plants, a lot of the treatment systems just disinfect the water, make sure that we kill off whatever organism is in there, and that would then protect public health. The caveat, though, becomes, well, if you add too much chlorine, does that pose a public health risk? The answer is yes. And so we have to balance kind of those types of things. Are you convinced by the conclusions? How much time do we have? Are we over time?
No, you have like 15 minutes of discussion, and then we want to open it up to Q&A. So they can ask any questions about you or about your field of work or the research article.
Yeah, all right. Yeah. Sounds good. So after seeing all those results, do you guys believe some of the conclusions that these authors presented? And I'll go back to those conclusions. Are the pathogens in sediments a public health risk? How many do you think yes? Based on the evidence provided here. And let's go to the evidence. So the actual pathogens are here. So the pathogens, again, are Salmonella, SAL, and ENTV. We can see that we can compare whether or not we find it in the water column versus sediment A versus sediment B. Sediment A is the one that has always been inundated in water. Sediment B is the one that was partially inundated with water. So, what do you think? Do we see differences between viruses in the water and viruses in the sediment? We're talking ENTV here.
In regards to ENTV, not really. 6 to 5 from sediment A to sediment B. That doesn't seem like a tremendous difference.
Right. Okay. What about between the sediments and the water itself? I mean, obviously, it is true there are a lot of non-detects, so a lot of the circles, dashed circles here. But let's look a little bit closer here, and we can see now. I should say that these are log transformed units, so that these are orders of magnitude. So like even a 1 unit change is actually a lot, right? That's 10 times really. So here we have, in the water, we have very, very low, anywhere between 1 to 2 orders of magnitude, which jumps up to anywhere between 2 to 3 in sediment A and right around the same for sediment B. So comparing sediment B with water, they're probably overlapping. There's probably no difference there. But I would probably say that, yeah, there might be some evidence to suggest that at least for enterovirus, we're getting more associated with the sediment than we are with the water itself, right? So there's a little bit of evidence here. All right. What about Salmonella? Want to venture an opinion on Salmonella? We all know Salmonella is bad, right? You don't want to be eating.
Well, before we make opinions, I could say it's actually, I am not very familiar with this type of graph, so I do not know exactly how to read it.
Okay.
Or like what the symbols and the colors mean.
Sure.
Do you think it'd help us understand it a bit better? So before we make an
opinion. Yeah. So this is basically time. So they started in November, so this is time, and they sampled once a month. Okay, so November, December, all the way through December again. So this is the sampling time. This is the quantity, how much of either the salmonella or the enterovirus or whatever microbial indicator is present on a log transform scale. So log of, for it to be 0, you would have to have 1 colony on there, right? So log of 1 is 0. And then so for log of 1, then you'd have to have 10, and so on. So 10, 100. We can form that back to the non-log scale. And then the different symbols, triangles here are the salmonella. The circles are the cultivable E. coli. And then we have opened and closed to represent the dry and the wet sampling. Down here, the diamonds are the ones that are enterovirus, and the coliphage are the ones that are the squares. So that's where that occurs. Having known that, does that help inform your opinion a little bit more? Yeah, so basically, one thing that you probably recognize is that there's a difference in scale between the sediments and the water column, especially for the salmonella and stuff, right? So here we don't go below 0, but here we get to 10 to the minus 2 on the scale. So it looks to me, just not really looking at the actual number itself, and just by looking at trends here, that salmonella is probably more in the water than it is in the sediment. Probably have higher concentrations here. Here we're looking between 1 to 3 orders of magnitude. Here we're looking, depending on if it's wet or dry, but mostly we're below 0, and then around 10 to the 2. And then in sediment B, it's just pretty much at 0. Right? So probably we might have more salmonella in the water. So personally, I think that probably does not suggest it as a good, this differential distribution between water and sediment for salmonella. That's probably not strong evidence. And then that's probably also why they didn't really go into much detail about that particular thing. All right. What about this idea that the indicators themselves are good at what they do to identify a pathogen, the presence of a pathogen? So here, again, in that shaded region is the indicator, the non-pathogenic indicator, and then the data below it are the pathogens, salmonella and enterovirus. Do you think that indicators here do a good job of what they're supposed to do?
I think maybe for salmonella it looks a little bit better as opposed to the virus, because for the virus there are the little dotted circles, so you can't really tell if there's something there.
Yeah.
But for salmonella, you don't really see that as much, and you can see that the black dots do correlate with the little triangles to indicate that there is salmonella in the sample. So.
No, you bring up a very good point here. You bring up a point where a lot of the enteroviruses are not even there, right? And so that kind of speaks to how sensitive is that indicator, right? So is there a minimum amount of enterovirus that needs to be present in order for to see a representative increase in the indicator itself, the coliphage? Here the coliphage there's not a lot of variation, right? And the coliphage is pretty flat. Here there's probably a little bit more variation. You see that there's a wider distribution, and here there's much more more variation because the distribution is very large here. But even when we have barely any enterovirus, we can't really have this kind of correlation between the amount of enterovirus there and the amount of coliphage there. So claiming that it's a strong indicator or a strong, performing indicator of, at least for enterovirus, may not be completely accurate. But there's a caveat to everything, right? So you can think, oh, well, this is just enterovirus. Maybe there are other types of viruses present that we're not measuring that's going to account for why there's such elevated risks here. So in terms of study design, you can see there are limitations. And there's going to be limitations with every study that you look at. And that's one of the things that we do in graduate school. We look at all these papers, we look at all these studies, and we're trying to identify what's a good study from a bad study. What study would help me create my own study, right? So seeing something like this, you have to be able to recognize, oh, that may be a limitation of this particular article that's, kind of discounts some of the conclusions that they came up with. They also claim that TEC should be used with caution because it did not correlate with CEC. Let's see here. So here it's CEC is in the left-hand side. This is the cultivable E. coli. This is the total E. coli. This is sediment samples. Let's look at the water samples. Yeah. Any thoughts on that? Do you consider these to be different enough? On the log scale, because it is log scale, that means that these are, these are very, very large differences actually between, say, 10 to the 9th and 10 to the 10th, right? We're talking about billion versus 10 billion. So, like, on the log scale, there are really big differences here. But one of the challenges you might, observe when you look at data like this is that there's huge variation, right? These are box plots, and you probably are not familiar with the box plot, but the box plot here is plotting the 25th percentile to the 75th percentile. So, 50% of the data are represented by the box plot itself. If you have a large box plot like this, that means there's widespread in that 50% of data. Whereas here, they're pretty much all, pretty accurate and consistent around 10 to the 9th. On top of that, you have large error bars. And this line here represents the median count. So basically just the middle number from that number of samples. So you can see that there are issues with comparing something where there's very tight distribution versus something where there's very large errors and large distributions, especially here. So to be able to say with confidence that there are clear differences becomes a little bit more challenging. So this particular article was, a great topic area, but when you look very closely at the results, they may not be as refined as you might have hoped. All right, that's pretty much the gist of it all. If you guys have questions about anything regarding, the path through graduate school or the article itself, I'd be more than willing to answer them or discuss them. Yes?
Did you ever consider another aspect of public health other than environmental health?
I've always been pretty much environmental health. Did not really look, I've been exposed to some of those things. Like we did have to take different types of public health classes going through the program. But, it's such a broad area that you can have areas that you just really don't really connect with at all. And you can have areas that you really are focused on and really connect with you. So that's, with me, it's always been the environment. It's always been water. So that's kind of where I stuck. But you do find your own path. You're depending on the program that you enter, there could be some flexibility. Sometimes they have these, if it's a lab-based program, you might have a lab rotation where you try out different things throughout your first 1 or 2 years. And then when you find that thing that sticks with you, you really grab onto that and focus on it. Yeah.
Do you do any work with policy when it comes to environmental health?
I currently don't do a lot of policy work, but I am involved in different types of research that may influence policy. So, and some that are very policy oriented already. So right now, the Southern California region, a lot of the water agencies in the Southern California region are worried about some of the EPA rulings that are coming from above. So the EPA is thinking about changing their indicators. They're looking at replacing some of their major indicators with the colophage. And so because of that, there's all this, all the agencies are now having to learn the methods to quantify the colophage, and then they're also trying to validate the use of colophage as an indicator for fecal contamination. So there's work I'm doing with them in terms of monitoring. So we'd have people go out to the beaches, streams, take some water samples, and look to see what the levels of colophage are, and whether or not it relates to some of the current standards, and if they're better than the current standards. So in a sense, it's more of a confirmation than it is of new policy. Yes.
What was the hardest part for you in terms of undergraduate school, graduate school, and like the different things you've learned pertaining to where you know?
Yeah. So with undergraduate work, I probably had not a unique experience, but I probably had a not so common experience where I thought about graduate school so late, so it almost like fell into my lap. But so the biggest challenge for me was really developing a support group, a network, and also connecting with faculty. So I don't know how often you guys actually interact with faculty, but it was in the last year of my undergrad studies, I did a research class with a faculty member who really was involved in everything that, in all this water research, and it just, she really guided me in the right way. So finding that kind of mentor really helped me pursue graduate school and, actually make my way through graduate school, because there's so much in graduate school where you may just get lost. It's almost like undergrad again, except a little bit more focused.
Any last questions?
Well, this is not exactly relative, but in consideration of like how much you study with water, so I was wondering if in your daily life, if it affected or influenced you in your choices of what kind of water selection you drink.
Oh yeah. So
let's say like a bottle of Lake Arrowhead versus Boss water. Sure. Like, does that influence you in some way?
No, absolutely. Actually, it's a good question, because you think about all the different waters that are out there, and what are the ones that are going to pick. For me, I actually am a fan of drinking regular tap water. I don't mind drinking regular tap water. Now, we have very good treatment systems here in the United States, and so that's kind of one of the reasons why I'm okay with it. But it definitely, it depends on, there can be, there's so many factors that play into this, right? Do you enjoy the taste of that particular water? That could be a factor that changes everything. For tap water, it's fine with me, but if you're drinking bottled water, now we have all these other things like kind of to think about. At least for myself, I think about, okay, all the bottled water means that there's going to be a lot of plastic production, right? So you're going to increase plastic production. So now what are we going to do with that waste? Where's it going to end up? Are you guys all familiar with the Pacific Gyre, the garbage patch out in the Pacific? There's islands actually that's about the size of Texas of plastic material that are floating out in the Pacific Ocean, just because we are doing our best to deal with plastic waste, but we're not doing everything possible to deal with plastic waste. In fact, we all think that plastic is recyclable, and it is recyclable, and people do recycle it, but we do such a poor job of recycling it that we don't handle the plastic waste very, very well. So it's ending up ultimately in the environment. So that's something I think about. The other thing I think about is how are bottled water regulated versus how tap water is regulated. Bottled water, it goes under much less regulation than tap water. So the levels of safety are not in place for bottled water where they are for tap water. Granted, with bottled water, it's usually market driven, so you want to make sure you're not poisoning the people that you're serving. But there are no regulatory, or there are not as many regulatory hoops that they have to jump through in order to produce their product versus the water facilities that bring tap water to our homes. Our tap water is disinfected, so we have a way to make sure that there's no bacteria or viruses in our water. But with tap water, disinfection doesn't always happen. It maybe happens once every so often that they identify a problem. So there are a number of factors that I think about what water I choose, but I'm completely fine and happy with tap water. It also costs thousands times less than bottled water. If you were not aware, if you compare the cost of a glass of water from the tap versus bottled water, you're talking about 1,000 to 2,000 times more difference. And of course, we're talking about parts of a cent here, but it adds up and it's pretty significant. Yes.
Do you find that your work, well, I guess environmental health, correlates or meshes with epidemiology more than like?
No, absolutely. It really does. I mean, when you think about identifying an indicator, right? That indicator has to have a relationship with a health outcome, right? And so what I would like to eventually incorporate into some of my research is validating that validating whether or not the indicator is good or bad with how it relates to some certain health outcomes that are being found. So you can see like maybe doing a survey with some of the surf rider organizations or the beach going organizations and figuring out how many people are getting sick. And then does that relate to some of the levels of indicators that we're finding? So there's definitely overlap there. And we call that quantitative microbial risk assessment. And so that's where this kind of complementing areas really work well together.
Yeah, because when we were discussing about the salmonella, then what came to mind was the recent salmonella outbreak with romaine lettuce.
Yeah.
And just like, obviously the lettuce needs to be washed before it's packaged. So maybe like looking at that link and seeing, is that something that relates to why there's a current outbreak right now, or just seeing those factors. It's really interesting to me.
Yeah, and there's so many lines along the path that really kind of intersect and may influence whether or not there's a contamination event. And so being able to study those numbers and that data is critically important. Yeah.
Any of the students have any more questions? I have a question, unless anyone students are ready. Okay. For drinking water, do you know a lot? I know tap is fine, but like, do you know a lot about whether or not like it's what the healthiest drinking water would be? Like, does it have to go through, or would it be recommended that it goes through some kind of thing?
Yeah. So with drinking water, there's several levels of treatment that occurs, and we do a great job here. Now we always do the proper treatment here where we filter things out. We make sure that all the particles are gone. We don't allow water to leave the drinking water treatment plant if it's in excess of small particles. We make sure so there's filtration that occurs to make sure that that's not a problem. And then eventually we even disinfect it. So that also makes sure that there's no problem with our drinking water. And they all go through that kind of level of stringency. They all go through different treatment strategies. There's not one, they don't really vary too much in practice because they're all required to do these 4 different steps before it can be to come out of our tap. Yeah. Okay. Yeah. But there's a bunch of different things we could consider too, like the actual source of water. Is it coming from an aquifer that's clean, or is it coming from, say, a river that is marginally polluted? So the source water quality can also have an impact on whether or not we're drinking safe water. The whole issue with Flint, and you're all probably heard of the whole issue with Flint, is that they changed their water source, and then in that change of water source, they didn't quite, there were some people dropped the ball, didn't really kind of communicate some of the corrosivity of that water very well, and so that ended up leaching lead from the pipes, and people were getting exposed that way. So those kind of things don't happen very often. That's why I'm still a proponent of drinking tap water. But you got to recognize that there is human error in certain things, and so it's something that I think about as well. But in terms of like your question about different drinking waters that are more safe than others, they're all pretty much on the same level. It's just really The only other thing to differentiate that is the sources. Here in Southern California, we get most of our water from the groundwater, as well as some imported water sources as well. I mean, if you guys have driven to San Francisco, the State Water Project, you passed it, that big canal of water that you see on the side of the road. That's the State Water Project, so they're bringing water down from the delta. And then we also take water from the Colorado River. So there's a Colorado River aqueduct that flows from the Colorado River and brings water to us here. So there's a lot of different water sources, and they're all, the ones here are all fairly safe to consider.
Well, thank you so much.
Yeah, sure.
Thank you.